Good evening, all. So I will wait for like another 2 to 3 minutes, and then we can start. And if you guys have any questions or doubts from the last class or the class before previous classes, you can ask me. By the time we are waiting, you can ask if you have any. Okay, so yeah, good evening, everyone. I am Srijan Sen, and I will be taking your live class session on the course Introduction to Experimental Biochemistry. Sorry, Introduction to Complex Biological Systems. Okay, which is being taken by Professor Shomode and Professor Dibyendu Samant of IIT Kharagpur. Okay, so I'm sharing my slides with you all. So usually, like I do, first I will be going with a brief lecture on the week courses or whatever was there within the week, and then we will discuss the question and answers, a few interesting question and answers in the end. Right? Okay, cool. Yeah, great. So today's class or the week 3 class was mostly on proteins. So proteins, if we tell in short, are the nanomachines of living systems. And it's a very interesting term, nanomachines of living systems, because this is the driving force, or the different functions of our body are taking place because of proteins. And proteins are a particular state which gives justice to whatever information is being stored in the DNA. You can say the DNA then transcribed into RNA and then translated into finally proteins. So proteins is the final stage or the machinery through which the different functions are carried out. So before we go into proteins, we need to understand what are the basic unit that builds up a protein, and that's an amino acid. Most of you might be knowing what an amino acid is. Those who don't, it's because it has an amino group and a acid group. Okay, so there is a chiral carbon. We can call it a chiral carbon because for most of the amino acids, the chirality is being maintained except for one amino acid, which is the glycine. Okay, so what's a chiral carbon? A chiral carbon is one that has 4 different functional groups attached to the 4 hands of a carbon. We are aware of it. Okay, and so in one hand you have the hydrogen and the R group. So R group can be different types. There are different types of R groups that can be present. So based on that, there are different types of amino acids which comes up. The other two hands are basically the amino group and the acid group. So based on the different types of R groups, you can categorize them into 20 different types. Okay, so individually there are 20 different amino acids, but now you can categorize them in different sections based on whether they are negatively charged, whether they are positively charged, if they're uncharged, if they're polar, non-polar. So different categories you can basically set them into, right? Now if we consider about the negatively charged amino acids, you have the aspartic acid and the glutamic acid. The positive charge includes the arginine, lysine, histidine, that mostly contributes to the uh, pushing the pi of the particular protein towards a higher region. Then you have the polar amino acids, the acids, the the amino acids that tend to be, or the residues that tend to be on the surface and water loving. Then you have the non-polar amino acids, like mostly those form the hydrophobic core of a particular protein and are mostly involved in the cation pi charge interactions. So there are different interactions that happens in a protein. We will come to that, but these are the categories of the different amino acids that are basically present based on the different groups that will be there in that particular R. Now, some basic chains that I was talking about are the positively charged amino acids. Why are they called positively charged? Because at physiological pH, they have a positive charge. Like for example, the lysine and the arginine or the histidine have the hydrogens that, so you see, so this group is basic because its positively charged is mostly stabilized by the resonance. Okay, and apart from that, these nitrogens have a relatively weak affinity for the hydronium ions or whatever you say, and only partly positive at a neutral pH. Now, at a physiological pH, these hydrogens are not exchanged and they tend to give positive charge to these particular amino acids. Similarly, for the negatively charged amino acids at physiological pH, they do not have the hydrogens attached and are negatively charged, hence we call them as the acidic or the negatively charged amino acids. Now, mostly the uncharged polar amino acids like the asparagine, glutamines, which are the amine, you have the amino groups that is present instead of the OH group over here, and that's asparagine and glutamine. You have serines, threonines, and tyrosine. So most of these have polar groups attached in that R position or that alkyl aline position, and these are mostly the surface residues of most of the proteins, because when we look into an amino acid, it makes no sense if it does not go on to form a protein. Okay, so whatever it is, so these are mostly the surface residues that are present over there. So polar side chains interacts well with water and can form hydrogen bonds. There are multiple non-polar amino acids because of their non-polar side chains. The basic structure is mostly formed by the carboxylic acid CO. You have the amino group, and in the middle you have a C alpha. That's the chiral carbon of the particular amino acid. So this forms the backbone. But mostly it's polar or an acidic or a basic amino acid because of its side chain. Good. Okay. Now, nonpolar groups are mostly unfavorable, are energetically unfavorable because they cannot be in contact with water and hence forms the hydrophobic core of the amino acid, and leads to hydrophobic interactions. Okay. Now you have two to three very interesting amino acids, though, that is tryptophan, phenylalanine, and tyrosine. So these amino acids are responsible. So because of their aromatic group that is present over there, these amino acids are responsible for providing absorbance to a particular protein. Like the more the number of aromatic amino acids you have in a particular protein, it gives to a higher molar extinction coefficient with a better absorbance required for tracking during purification or downstream purification processes. So these amino acids are basically important because they absorb at 280 nanometer, and that's how you can track a particular protein if you're measuring the absorbance of proteins at 280. So aromatic residues are responsible for the absorbance of particular proteins. We need to just keep these things in mind. Three important residues: tyrosine, tryptophan, and phenylalanine. Now, amino acids are linked together by the covalent peptide bond. So there are two amino acids. You have an NH3 plus group and a COO minus group. So there is a loss of water, and a peptide bond formation happens after the loss of water, and you get whatever amino acids are there one after the other. So you have a glycine followed by an alanine over here. So peptide bonds are formed by the loss of water. Now then there can be multiple amino acids. So multiple amino acids comes one after the other with the loss of one molecule of water. So if there are two amino acids that is forming a peptide bond, there will be loss of one water molecule. If there are three, it's n minus one if it is for a linear protein sequence. Now if you require a cyclic protein or a cyclic peptide, whatever it is, then there will be loss of n number of water molecules. So n number of water molecules lost is equivalent to n number of peptide bonds formed in a cyclic peptide or a protein. But for a linear one, there should be, for n number of peptide bonds formed, or for n number of amino acids, there should be n minus 1 number of loss of water molecules. So one molecule of water lost is equivalent to one peptide bond formed. And that is what is being shown over here. What comes after the other does not matter, but the entire concept is a peptide bond formed by the formation by the loss of one molecule of water. Now it's very important to understand the characteristics of the polypeptide synthesis. So the amino group of one amino acid reacts with the carboxyl group of the other to form a peptide bond, also known as the amide bond, amide linkages, with the elimination of one molecule of water. Now, the polarity of the water molecule is the polarity of the molecule is retained. So the monomers are all joined together in the same orientation in which they are coming, and the polypeptide chain has a polarity. Now, the polarity is generated because the all the amino groups are pointing in one particular direction, and all the acid groups are pointing in the other direction. So the amino group that is pointing in one particular direction has a positive charge. The acid group has a negative charge. So there is a polarity that happens in a particular uh, polypeptide chain, and the amino side, or where the amino groups are actually saturated, that's called the N terminus, because it's an NH3, so an N terminus, and the acid group which has a CO minus is called the C terminus of a particular polypeptide chain. So the polypeptide contains a free amino group at one end known as the N terminus, and the free carboxyl group at the other end known as the C terminus. Now these ends of polypeptide chains are referred to as the amino and the carboxyl termini, or in short N and C terminus respectively. Now the product of the condensation reaction preserves the reactive group, the carboxyl group at the end of a molecule. This makes it possible for the polypeptide chain to join to yet another amino acid. So there can be an elongation one after the other, so these ends are quite open. If so, this becomes quite important, or quite interesting when you're doing a chemical synthesis. So for these are open ends which we call. Otherwise, if you need to stop a reaction at one particular point, you have to use some moieties, like there are different groups, blocking groups, for keeping the free amino and the free carboxylic group blocked. Okay. Now, so polypeptide chains are one after the other, where the amino acids are attaching, is called the primary sequence of a protein, right? But proteins do not exist. So the basic difference between a polypeptide chain and a protein is basically there is a fine line of difference. So polypeptide chains have its joining of sequence one after the other does not matter how the sequence aligns, how what happens, it does not matter. A protein is a functional unit. Okay, it is also made up of amino acids one after the other, but it has a folded structure, it has a function, as well as there are certain groups of amino acids that folds in a particular orientation, giving it different folds. 2D structures, 3D structures, quaternary structures are formed for a particular protein, which is not required for a polypeptide chain. Now, there are different 4 levels of organization of a protein structure. You have a primary structure, that's the joining of the amino acid residues one after the other. Now, these primary structure leads to a secondary structure. There can be multiple secondary structures, like alpha helices, beta strands, beta sheets. Then you have the different folds, turns that can happen. So these are the secondary structures. One or more secondary structures gives rise to a tertiary structure, and finally multiple tertiary structures aggregate together to give a quaternary structure, or sometimes there are multiple monomeric units of the tertiary structures. Polymeric orientation of the tertiary structures for the function, so therefore they have to get to the quaternary structure to carry out their activity. Okay, so the primary structure of a protein is a sequence of amino acids linked together by peptide bonds, that is the covalent bond. And the secondary structure of proteins where hydrogen bonding is involved, that's the polypeptide forming into alpha helix, beta sheet, random coils, like you have the turns, folds. Then the tertiary structure of a protein is the three-dimensional folding of a single polypeptide chain where you have the hydrogen bonding, disulfide bonds, van der Waals interaction, ionic bonds, electrostatic interactions, hydrophobic interactions. Okay, so these interactions are responsible for the tertiary, for the maintaining the tertiary structure of a particular protein. And the quaternary structure of proteins is basically the association of two or more folded polypeptides or the three tertiary structures of proteins to form a multimeric protein. That's the bonds and interactions similar to the tertiary structure. Good. Okay. Now the unity and diversity of proteins. So there are multiple types of proteins that can be present. Some works as a monomeric unit, some works as a polymer because they require its other partner proteins to work like that. Okay, so the primary structure is the sequence of amino acid that determines the structure of a particular protein. So for example, you have a purified protein which is isolated from cells and you are exposing it to a reducing environment. Okay, that's a high concentration of urea. What happens? The protein gets denatured. What is denaturing of proteins? Denaturing of proteins does not affect the covalent bonds that are between the proteins. It just breaks apart the weaker bonds. It provides the protein a reducing environment where the disulfide bonds basically break. And once the disulfide bond breaks, there is mostly the protein opens up and it denatures. Now, after the denaturing of a particular protein, its activity is lost. Now, when you place that protein again after removing of urea in a buffer condition where there is no reducing environment, rather you do not have to provide an oxidizing environment, but if you're just removing the reducing environment, the protein reforms and gets back to its original conformation in which it actually was. Now, in water, hydrophobic amino acid cluster inside a folded protein away from a solvent. Why is it so? So you see, protein is mainly formed of polar amino acids and the non-polar amino acids. Now, polar amino acids are basically water-loving. Non-polar amino acids do not love water. So, a protein is a combination of multiple types of amino acids. The folds of the protein is determined not only by the structure but also by the choice of the amino acids to be either away from the solvent or towards the solvent. So mostly the non-polar amino acids are forming the core or the hydrophobic core of a particular protein that is mostly responsible for the holding the protein to its right structure. And the solvents that are the polar amino acids are responsible for the different kinds of activities by interacting with water, hydrogen bonds, and are responsible for holding the outer surface of the water. So the hydrophobic core region contains the non-polar side chains, and the hydrogen bonds that can be formed is via the polar side chains on the outside of the molecule. So the polar amino acids forms the surface residues. Now that's about the primary structure, but as I was speaking, the primary structure is not the ultimate structure. It forms multiple secondary structures. So the secondary structures of proteins, the first one comes is the alpha helix. So hydrogen bonds between the NH of every peptide bond and the CO of the next peptide bond of the chain. So it's not the next peptide bond, it's N and N plus third, N plus 4. So the amino group of nth amino acid forms a bond with the N plus 4th amino acid. Here the R groups are not involved. What is involved is basically the hydrogen of an amino group and the oxygen of the carboxylic group. And when the alpha helix forms, so it's like a spring-like structure. So you can see over here that when the spring is formed, there are multiple amino acids that are arranged to give it a helical structure, and the pitch of this particular alpha helix is basically 0.54 nanometers. This the other one is the other secondary structure that can be formed is beta sheet. So polypeptide chains are held together by the hydrogen bonds over here, again formed between the NH group of one polypeptide chain and the CO group of the other chain, right? Now over here also you will see that there can be multiple types of beta sheets that can be formed. One are parallel beta sheets, one are anti-parallel beta sheets. So you see that if it's a parallel beta sheet, you see over here it is a bond formation between the NH group of one particular amino acid with the CO of the other, whereas it's an anti-parallel beta sheet which is more stable because the distance between the hydrogen bond formation is much less. So the distance between the H and the oxygen is much less, so the formation of the stability of the hydrogen bond is much more for an anti-parallel beta sheet rather than a parallel beta sheet. So alpha helices can wrap around one another by interactions between the hydrophobic side chains to form a stable coiled coil structure, which happens mostly for alpha keratin in the skin and myosin in muscle. So there can be multiple other structures as well that can also form like that. And what are folds and turns? So folds are basically they do not form any structure. It's a very linear structure, but just because it is responsive, the folds are because, or turns are because you need some residues to give a kink. So for example, if you have a beta sheet and you want a turn over there, you would probably use one particular beta turn, and beta turns are quite rich with one or two prolines, because prolines are very rigid amino acids. You might be knowing they have a very rigid structure. So these are responsible for providing bends or kinks when you are attaching two different beta sheets. Okay, and turns are mostly you will see for different transcription factors or intrinsically disordered proteins. They do not have well folded structure. They are like a very flexible structure, dynamic structure, so they do not have any secondary structure. They have mostly have the folds. Now the tertiary structure of proteins is basically the 3D conformation or shape. So it depends on the properties of the R groups of amino acid residues. They fold spontaneously or with the help of molecular chaperones, and they are stabilized by covalent as well as non-covalent bonds. So many proteins are composed of separate functional domains like bacterial catabolite protein, the cap proteins, then the protein domains. Um, so there are different functional domains that are present in proteins, and these domains are segments of polypeptide chain that fold independently into stable structure. So like you have an alpha helix, you have a beta sheet, but you see so a combination of multiple alpha helices and beta sheet gives you a particular domain. Now there is a difference between a motif and a domain. So if it is basically it's preserved or conserved over a generation to generation, you called it a domain, right? Now non-covalent bonds helps in protein folding. So there are multiple non-covalent bonds, like you have the ionic bonds, hydrogen bonds, van der Waals interactions, hydrophobic interactions are that play a very important role in the protein folding. So no, it's not only that only covalent bonds have to form, like you have the peptide bonds or the disulfide bonds. Now the covalent disulfide bonds happens between the adjacent cysteine side chains. Okay, and that also in an oxidized condition. So if they're in an oxidized condition, then there is interchain disulfide bond. There can be intra-chain disulfide bond. Intra-chain disulfide bond is important for maintaining the structure of the protein. Interchain disulfide bonds are responsible for forming polymeric structures of proteins, things like that. So for many plant proteins, you will find that there is an oxidized condition and there are multiple cysteines that are present in these proteins, and that maintains, so the cysteines maintain the structure of that particular protein. But for other cases, you will see that there are polymeric structures that are also forming due to inter disulfide bonds as well. So these are the types of covalent bonds that are present. And when you place these proteins in a reducing condition, the disulfide bonds break, the proteins open up, and the activity of the these proteins, or if these are enzymes, then it's actually lost. The molecular chaperones are proteins that helps in the folding of other proteins. So these are also proteins, but it helps in the folding of other proteins by the use of ATP. Now, how is the tertiary structure of a polypeptide chain stabilized? So you have ionic bonds. It's between a charged amino acid chains at a cellular pH of 7, and then it can be between lysine, aspartic acid, arginine, glutamic acid, and there can be many permutation combinations, but ultimately between a positively and a negatively charged amino acid. Now, hydrogen bonds between the R groups, like you have the serine, threonine, tyrosine, that can also occur with surrounding water molecules. There are covalent bonds, like the disulfide bonds between the cysteines. You have hydrophobic interaction, that's the polar side chains associate in the interior of the molecule and forms the core of the protein. And then the van der Waals interactions, quite weak interactions, but multiple van der Waals interactions play important role, right? Now, finally, we have the quaternary structure of proteins. That's hemoglobin, protein in red blood cells, has four subunits. So, two of alpha and beta globulins containing the heme molecule. So, individual these are the proteins, but for the hemoglobin to be active, it requires four subunits altogether. So, individual units are not active. Okay, so the four units together are responsible for containing the heme molecule. Okay. Any questions up to here? No questions? Okay. So now, now we need to understand that. So we talked about the structure of proteins, and structure of a protein determines the function of a protein. So there is a protein folding problem. So what determines the structure? The energy, kinematics, these are responsible for determining the structure of proteins. Now, how can we determine the structure? There are multiple experimental methods, computational tools. Nowadays, there are so many developed AI tools, AlphaFold, that can help in the understanding the structure of proteins. Previously there were like NMR, like we still have NMR, but that's for the solving the structure. It's quite simple nowadays with the level of computational improve development research has found. And then you have the molecular structure that are responsible for the function, where it plays role in the regulation, then movement, catalysis for the enzymes, transport channels. There are multiple protein signaling. Okay, so protein folding is basically quite a problem, but it's important as well. So the amino acid sequence of a protein determines the 3D fold. That was done by Affinson, or the Affinson's experiment. So there are some exceptions, that all proteins can be denatured. Some proteins can have multiple conformations. So one particular conformation of a protein might not be there, but that's not a problem. Some proteins get folding help from the chaperones. So chaperones are proteins that help in the folding of other proteins by the use of ATP. So the function of a protein is basically determined by its three-dimensional fold. So can we predict the 3D fold of a protein given its amino acid sequence? Obviously, why not? Because when we know the primary sequence of a protein with, with the number of tools that are available nowadays, it's possible. Now, for example, you have a protein that assumes three possible conformation per peptide bond. So there can be 3 into 10 to the power 100 different conformations. That is around 10 to the power 47 conformations. So if there is a fast motion of around 10 to 15 seconds per sampling of all the conformations, it would take around 10 to the power 25 years. But each protein folds so quickly into a single native's conformation. It takes quite like nanoseconds or even femtoseconds for a protein to fold. So what is this? So that's where it's the Leventhal paradox. So if we look into the overview of the energetic contributions, so there can be different types of non-covalent interactions. That's the hydrogen bond that contributes about 3 to 7 kilocals per mole. You have ionic interactions, attraction, repulsion, that counts for around 10 kilocals per mole. You have hydrophobic interactions around 2 kilocals per mole, and van der Waals interaction around 1 kilocals per mole, with some covalent interactions like the disulfide bridges around 51 kilocals per mole. Now for the assisted folding of proteins, not all proteins fold spontaneously as they are synthesized in the cells. So there are chaperones that are present in the cells that help in the folding of many proteins. Okay, so you have the class 1 chaperones like the heat shock protein 70 type of chaperones, HSP70, HSP40, DNA-K, DNA-J. You have the class 2, that's the chaperonines. You have the GRO-L, GRO-S. Okay, then you have other enzymes that are involved in protein folding, like the protein disulfide isomerase, protein propyl cis trans isomerase. Okay, so these are the different types of chaperones that assist protein folding. So this is about the protein folding, but there are also conditions where a protein denatures. So the conformation of a native protein is marginally stable. The conditions are different from those in a cell, therefore it can cause different structural changes, loss of three-dimensional structure. So the loss of three-dimensional structure leads to the denaturation of the protein. The state of, or the condition in which half of the temperature in which half of the protein is, or half of the structure is lost, or the percent unfolded, the percent 50% of the protein is unfolded, you call it the melting temperature of the protein. Now in a denatured state, the conformation of the protein need not be completely randomized. So there can be number of physical and chemical agents that can cause protein denaturation. One example is basically heat, in which the complex effects on multiple weak interactions in a protein, that's basically the hydrogen bonds, or interactions that are weaker than 10 kilocals per mole maybe. So on heating, a protein's conformation generally remains intact until an abrupt loss of structure occurs above the melting temperature. So the abrupt loss of structure suggests a cooperative process in which the structure in one or more protein rapidly destabilizes the structure of the other parts. And this can also happen when you're adding reducing agents. So proteins are also denatured by the extremes of pH, miscible organic solvents like alcohol and acetone, by certain solutes such as the reducing agents like urea, guanine hydrochloride, or by the use of detergents. So detergents, what they do, detergents are charged molecules. They add charge on top of the amino acids, and that leads to the repulsion and opening up or denaturing of the protein. So none of these agents breaks the covalent bonds, because covalent bonds are strong bonds. So organic solvents like urea and detergents act by disrupting the hydrophobic interactions that produce the stable core of globular proteins. Urea and guanidine hydrochloride breaks the hydrogen bonds, and extremes of pH alter the net charge, causing electrostatic repulsion and disruption of the hydrogen bonding, which also happens when there are detergents present in the solution. So the denatured structures resulting from these various treatments are not necessarily the same, and not all proteins denature at the same conditions. So we need to be also aware of that. So lastly, most uh, proteins aggregate, precipitate out when they are in the denatured state, and that's quite common, and it's difficult to get them into folded state even when they are the reducing or the extreme conditions are removed. Once they precipitate, it's gone. And we also need to be aware that once when a protein starts precipitating, it also pulls the other molecules of the protein to precipitate out. Right. Now, renaturation of the proteins. So, early studies that were examining the renaturation of the refolding of protein of an enzyme ribonuclease provided experimental proof that tertiary structure of the protein is determined by its primary structure. So, what in this experiment, what was done? The purified ribonuclease was denatured, and by exposure to the concentrated solution of urea and a reducing agent mercaptoethanol. Now this unfolded the protein and reduced all the disulfide crosslinks to the cysteine residues. Now when urea and mercaptoethanol were removed, the randomly coiled denatured ribonuclease spontaneously folded to its catalytically active state. Now even the protein's 4 unique disulfide bonds reformed correctly. And this experiment provided the first evidence that the amino acid sequence of a polypeptide chain contains all the informations that are required to fold the chain into its native three-dimensional structure. Now, there is a thermodynamics behind this protein folding. So, thermodynamically, you can look at the protein folding process as a kind of free energy funnel. So, the unfolded states mostly remains. So, these are high energy states, and these are uh, unfolded states that are characterized by the high degree of conformational entropy and high free energy. Now, as folding proceeds, the narrowing of the funnel reflects the decrease in the conformational space that it searches for in its native state, and there are small depressions that you can observe at the sides of that energy funnel, that is due to the local uh, folding, like the folding of the secondary structures, the generation of the secondary structures, and finally when the tertiary structure forms or the folding process is complete, which is definitely a slow process, it reaches the bottom of the funnel. So these are like the semi-stable intermediates, and from when it again unfolds and gets back to this native conformation. So that is the conformation which we expect. So you see over here the entropy is very high. These are the semi-stable states, and after it reaches a particular value, you see the native structure forming, and this is called globular proteins form, because these are properly well folded proteins with low entropy and lower energy values. Any questions? No questions? Okay. Then we will talk about the protein ligand interaction. So, in the 25th anniversary of the Institute of Molecular Biology in the University of Oregon, Linus Pauling said that the secret of life is the molecular recognition. That's the ability of one molecule to recognize another through weak bonding interactions. So, binding is the first step necessary for a biological response. So before macromolecules can perform a function, they normally just interact with a specific moiety, which we call as the ligand. Now, in some cases, like you have myoglobin binding, is a subsequent release of the ligand that which might be the sole function of a macromolecule. But to understand binding, we must understand the equilibria that's involved between them, how the binding is affected by the ligand and the macromolecule concentration, and how to experimentally analyze and interpret the binding data and the binding curves. So reversible binding of ligands, which is essential because the specificity of the ligands and the binding sites needs to be understood. Ligand binding is often coupled to conformational changes, which is sometimes quite dramatic, which is induced fit or in multi subunit proteins, conformational changes is on the subunit which can affect the others. That is cooperativity, which happens for hemoglobin because there is a cooperative effect of the four subunits that are associated with it, and it should be made sure that the interactions can be regulated. If there are uncontrolled interactions that goes beyond, which happens most of the time, that the interactions are too strong, it can affect the downstream signaling pathways because most of the interactions happens are associated with multiple signaling pathways, and that can lead to cancerous conditions, cellular proliferation, stuffs like that. So for the interaction with other molecules, it should be made sure that it's a reversible transient process of chemical equilibrium. So the molecule that binds the protein is called a ligand, which is a small molecule, not necessarily, can be other proteins, carbohydrates, then peptides, small molecules, can be multiple things like that. Now, a region in the protein where the ligand binds is called the binding site. The binding site may not be the same as the active site of the protein. We need to be cautious about the terminologies because the binding site is where the ligand binds. The active site is what is responsible for the catalytic activity of the particular enzyme. Now, the ligand binds via same non-covalent forces that dictate the protein structure. It's nothing different than that. There are hydrogen bonds. There are van der Waals forces as well. Sometimes hydrophobic interaction, cation pi interactions, stuffs like that. So binding is a quantitative description. So if we consider a process where the ligand binds reversibly to the site of a protein, so it's the rate of association and kd is the rate of dissociation. So the kinetics of such a process is described by the association rate constant or the dissociation rate constant. And if we want to write the equilibrium equation, we can write it as the Ka, concentration of protein and concentration of ligand, which is equal to the dissociation constant multiplied by the concentration of the complex. Now the equilibrium composition then gets characterized by Ka, the capital Ka, which is equal to the rate constant, association rate constant divided by the dissociation rate constant. Now, binding, which we mostly analyze as in terms of the bound fraction. So, in practice, we often determine the fraction of occupied binding sites, which is theta. So, theta is basically the total, the concentration of the complex divided by the total protein concentration, which is there in the concentration of the complex plus the unreacted protein. Now, when we are substituting the complex concentration with the previous equation, then whatever comes over here is entirely based on the ligand concentration. So theta becomes ligand concentration divided by the ligand concentration plus 1 by Ka. So 1 by Ka is basically Kd, which is the dissociation constant. So Kd value is basically, so you can get the Kd if you know what is the bound fraction terms. That is important because dissociation constant is basically, or the KD value is an indication of how strong your interactions actually are. Now, when you plot theta versus the ligand concentration, from there you can get the KD value. How can you get it? Uh, the ligand concentration in which 50% of the ligands have bound is basically the KD value. In terms of concentration, you get it, right? So the fraction of bound sites depends on the free ligand concentration and the KD. So experimentally, if the ligand concentration is known, KD can be determined graphically or via the least squares regression. Yeah, so KD can be determined graphically if you know the ligand concentration and the bound, the fraction of bound sites. So also you can graphically analyze KD from the slope if you are plotting the complex concentration divided by the ligand concentration versus the complex concentration. So if you are plotting it, you get a line curve, and from there the negative slope is 1 minus 1 by KD. From there you can also get the KD value. So binding has thermodynamic connections. So binding has certain thermodynamic connections. So interaction strength can be expressed as association or binding constant, that is K, which have units like mole inverse. Then dissociation constant Kd, which has units like molar. Then interaction binding free energy, that is delta G naught, which has units like kilojoules per mole. So by definition, you go delta G naught equals to delta H naught minus T delta S naught. That's, you get an idea about the enthalpy and the entropy. Then K, which is, and KD definitions are anyways over there. So from there, from delta G naught, you get the value of KD. How? By substituting all of the stuffs, you get delta G naught equals to RTL and KD. And lower the KD value, higher is the affinity for the protein for that particular ligand. So strong binding means KD value less than 10 nanomolar, and weak interactions mean KD value greater than 10 micromolar. So most of the protein ligands, very common protein ligands, are basically avidin, biotin, which is the strongest known interaction in 10 to the power minus 15 range, molar range. You have human insulin, okay, that's 10 to the power minus 10, that's antibody antigen reaction. You have specific protein DNA interactions, mostly for the transcription factors, receptor ligand interactions. Those falls in the quite a strong interactions. And for weak interactions, you have like the calcium calmodulin interactions, which falls in the low affinity, enzyme substrate interactions. So these are some of the weaker interactions. So the Gibbs energy of binding. So the binding constant can be connected to the Gibbs energy of binding, which I was just speaking about. That's delta G naught is RTL and KD. Okay, so you can neglect the sign as of now. It depends on how you're writing the equation. So the binding constant of 10 to the power minus 9 corresponds to a Gibbs energy of 53.4 kilojoules at body temperature. And from these equations, so these are the different equations. You don't need to remember all of that. What you need to remember is basically this particular equation will do, where delta G naught equals to RTL and KD, and that will do for most of that. And some important Protein ligand interactions are mostly governed by the hydrogen bonds, which are typical, untypical, charge resistant. You have ionic interactions or the electrostatic interactions that are due to the effect of the pK values, local pH of the binding environment. You have the cation pi interactions, then the hydrophobic interactions, which is mostly due to the displacement or liberation of water molecules from the lipophilic environment of the binding pocket. And if you need to check like for how you going to test the proteins, whether your protein is there or not, you can go for a sodium dodecyl sulfate polyacrylamide gel electrophoresis or an SDS page gel electrophoresis. Some people go for a native page electrophoresis, but these are the different ways of how you get the different protein bands. So these, every black spot is basically a protein band. So after you stain them, so first you use some running dye or a tracking dye to run the protein samples. You make the proteins negatively charged and you run the protein in this gel cassette. You run the protein over here, you have a positively charged electrode and the protein. So this is the direction of migration, and after, so they migrate at the different rates based on the size of the protein. Okay, and then when you stain them, they form black bands. Now coming to membrane proteins, membrane proteins are basically, mostly they can be the recognition proteins that have carbohydrates chain attached. So membrane proteins are basically present in the, associated with the cell membrane. So they can be the peripheral proteins you see over here, integral proteins, then lipid linked proteins. Some are attached with the cholesterol, phospholipids, oligosaccharides, glycolipids, things like that. So the carbohydrate chain, so the recognition proteins have carbohydrate chains that aid in cell identification. Like the red blood cells identifies the viruses as the foreign materials, and the other one is the receptor proteins that receives chemical signals from the other cells, such as the nervous system cells. Then there are channel proteins. So these proteins are also membrane proteins, and they help in letting in and out of certain substances of the cells. Like you have the aquaporins that lets in water in and out. You have the sodium gate channels, the potassium gate channels as well, that leads to the entry of the sodium and the exit of the potassium. So there are different gated channels as well. The two important types of proteins include the integral and the peripheral proteins. Like integral proteins cross both layers of the phospholipid bilayer, like you observed. These are the integral proteins that cross. So you have a polar group over here and the non-polar groups over here. The peripheral proteins are only on the top half or the bottom half of the phospholipid bilayer. So the primary function of membrane proteins include cell signaling. You have enzymatic activity, transport, which is important, cellular joining, cell-cell recognition, attachment to the cytoskeleton and the extracellular matrices. Now the types of integral proteins can be like so the type 1 is single transmembrane span, where the N terminus is the, you see the ectodomain, and the C terminus is in the cytosol. Or the protein can be entirely reversed, where the, you see the C terminus is in the ectodomain and the N terminus in the cytosol. Then you can have multiple spans of proteins that are spread across the two, the ectodomain and the cytosolic membrane. So that's the type 3. Type 4 have several different polypeptides assembled to form a channel. And the type 5 are lipid linked proteins. So this is the lipid that's linked to the protein, and the protein is present in the cytosolic environment. And type 6 is basically the proteins with both transdomain component and the lipid anchor. So you have a lipid anchor that is present in the biliary region, and the outside you have a part of the protein, and in the inside there is a hanging tail. And a part of the protein in the ectodomain is basically linked with the lipid membrane. Now, the properties of integral proteins are basically because of their hydrophobic transmembrane domains. The integral membrane plays a difficult role in getting these proteins in a soluble form once when they're out of the cellular environment. Now, removal of these proteins from the membrane requires use of detergents, ionic solvents, non-ionic detergents like the Tritonix, which generally does not alter the protein's tertiary structure. At the same time, it just releases the protein out of that bilayer membrane. Now, use of mild non-ionic detergents are mostly used for solubilizing, purifying, and the reconstitution of the functional membrane protein systems. Because at the end of the day, what you require is a pure protein to work with, to functionally characterize, structurally characterize, whatever you have to do, you have to work with a pure protein, and you have to get these pure proteins. Or if it, even if it's a membrane protein or a cellular protein, whatever it is, you have to get it in the pure form. Now, the transport of materials involves the passive transport that is mostly dealt by diffusion, simple diffusion, facilitated diffusion, osmosis, or an energy consumption transport like the active transport and exoendocytosis. Okay. Yeah. So if you have any questions, so that's all from my part about the lecture. If you have any questions, you can ask me. Okay, so if you have no questions, then we can go into the quiz time. Okay, so these are basically on you guys. So let's start with question one, then you can answer me in the chat box. I'm okay with that. Okay, let's start with the first four. Okay, one, two, three, four. So let's start with the first one. Which bond links amino acids together in a polypeptide chain? Quite easy questions. I want you guys to answer them. All of you try to answer. These are not very difficult. So, option B, yes, peptide bond, that is correct. Question 2, which of the following amino acid side chains are positively charged at physiological pH? So MSQ means multiple answers can be correct. MCQ is a single answer correct, but MSQ can be multiple answers correct. So I want all of you to give answers, not one or two people. These are not very difficult questions, and these are very um, important questions because these will be there in the exam. Most of you have seen this type of questions even in the assignment. So the correct answer is lysine, arginine, and histidine, ABD. Okay. Question 3. The polarity of the polypeptide chain arises due to random orientation of amino acids, altering R groups, presence of amino and carboxyl termini, hydrogen bonding. No, this question is not an MSQ, so it's an MCQ. So the correct answer is option C only, because of the presence of the N and C terminus. Question 4 is an MSQ. What statements about peptide bond formation are correct? So question 4 says which statements about peptide bond formation are correct? So what are the correct one? Yeah, A and B, because water is released during bond formation, and its loss of water during a bond formation is called the condensation reaction. So A and B are correct. Okay, let's go to the next 4. Question 5. Primary structure of a protein refers to. So what is the primary structure of a protein? It is defined as linear sequence of amino acids in a polypeptide chain, and this sequence is basically determined by the gene encoding the protein. So option C, which is the sequence of amino acids. So question 5 answer C. Question 6 says which interactions stabilize the tertiary protein structure? Hydrogen bonds, disulfide bonds, hydrophobic interactions, peptide bonds. So stabilizing the tertiary structure of proteins. It's an MSQ question, so yeah, hydrogen bonds are correct. You also have the disulfide bonds as well as the hydrophobic interactions that are responsible for the internal stability or the core stability of particular proteins. Right? Now, question 7. In an alpha helix, hydrogen bonds are formed between. So, how are the hydrogen bonds formed in an alpha helix? Yes, that is correct. NH and CO. No. It's not B, it's C. NH of one residue and the CO of the next residue. That is correct. So option C, question 7, that's C, not B. Okay. Question 8, which proteins are rich in alpha helical structure? Fibronine, myosin, hemoglobin. So over here, all the answers will be correct. A, B, C, and D. So all of them are rich in alpha helical structure. The question 9, that's beta sheets are stabilized primarily by disulfide bonds, hydrogen bonds, hydrophobic interaction, or ionic bonds, ionic interactions. It's an MCQ, so one answer is correct. So beta sheets are stabilized by, yes, that's correct. Oh, I got an answer from a new person. That's good. Thumbs up. Yeah. Hydrogen bonds between the polypeptide chains. Yeah, that is correct. Question 9, option B. Let's go to question 10. That's hydrophobic amino acids in aqueous solution tend to remain exposed to the solvent, cluster inside folded protein, stabilize tertiary structure, increase the entropy of folded state. It's an MSQ question, so more than one option can be correct. Yes, that is correct. Cluster inside the folded proteins, and when they cluster inside, there are hydrophobic interactions that stabilize the tertiary structure. Very good. B and C both are correct. Which level of protein, question 11, which level of protein structure involves functional domains? Primary, secondary, tertiary, or quaternary? That's option C is correct over here. That's tertiary structure. They have functional domains. Now, a very important point is what is the difference between a domain and a motif? Like I was telling, motifs are relatively small combination of secondary structural elements like multiple alpha helices or beta sheets with a specific geometry. Domains are much larger, they have an independent folding, compact globular region, and is basically a part of the tertiary structure element, right? And okay, and so multiple motifs can also lead rise to a domain. So anyways, so question 11, tertiary structure is the correct answer over here. Question 12, protein domains are characterized by independent folding, size of 1100 to 250 amino acids, requirement of chaperones, always structural stability. Okay, A, B, B, D. Yes, so structural stability is correct. Independent folding is also correct over here. So protein domains have an independent folding. They are structurally stable as well as they can be of a size of 100 to 250 amino acids. So A, B, D, all three are correct over here. Right, let's go to question 13. That is, which bond is formed between the cysteine residues? Very simple. So which bond is formed between the cysteine residues? A quick answer, hydrogen bond. Okay. You get a hydrogen bond between the cysteine residues? No, it's a disulfide bond. A covalent disulfide bond is formed between the cysteine residues because they have the SH group. In an oxidized condition, you get the disulfide SAS linkage. In a reducing condition, the groups are have the hydrogens attached, the SH group. Okay, so disulfide bond. Question 14 says which chaperones belong to class 1 family? HSP70, HSP40, GROEL or DNA-K. I spoke about the types of class 1 chaperones. In that I gave the example. So what are which chaperones belong to class 1? It's an MSQ, so more than one option may be correct. So MSQs does not mean always that there will be multiple options that are correct. It can be that one option is also correct, but it may be one or more than one may be correct. Okay, so be careful. Sometimes only one option is correct, but people tend to see that it's an MSQ and give multiple right answers, and that might turn out to be wrong. So class 1 types of chaperones includes the HSP70, HSP40, DNA-K and DNA-J. Okay, so over here A, B, D are correct, right? Okay. Question 15 says the Leventhal paradox highlights the problem of protein aggregation, protein denaturation, exhaustive conformational sampling, disulfide bond formation. Which of these does Leventhal paradox highlight? Okay, I've got one answer. It's not about an aggregation or a denaturation, it's an exhaustive conformational sampling. So the question still lies, how does, even if there can be multiple conformations that are available for a protein to fold, how does the protein know which conformation to actually have? And protein folding, for a protein to fold, it takes about quite nanoseconds and femtoseconds, but the calculation says it should be taking years. So that's the problem. So exhaustive conformational sampling. Now the next one, question 16, that's heat denaturation can be caused by heat, extreme pH, urea, breaking of peptide bonds. So how does a protein denature? So what are the conditions in which a protein can denature? Another MSQ for you. Breaking of peptide bonds is not denaturation. Denaturation means the primary structure of the protein is not lost. If the peptide bonds break, the primary structure is lost. So heat breaks the in the weaker interactions. You have extremes of pH that breaks the electrostatic interactions, and urea that gives it a reducing condition, breaking the disulfide bonds. So A, B and C are correct. D, breaking of peptide bonds, is not denaturation, that's something else. Okay, so protein denaturation does not mean the peptide bonds, the backbone of the protein breaks. It breaks the folds of the protein. Okay, or the interactions that are involved in the stabilization of the protein. Those are the ones. Okay, question 17 says which experiment evidence proved that primary structure determines tertiary structure? Hemoglobin structure, Aphelson's ribonuclease experiment, Leventhal paradox, or the gel electrophoresis? Which experimental evidence proves this? The Aphelson's ribonuclease experiment, in which first of all you had folded structure, you unfolded it and you get to a primary structure, and upon removing the reducing condition, it becomes folded. So the primary structure remembers which tertiary structure to form. So yeah, option B is the correct one. Okay, urea and guanidine hydrochloride denature proteins by disrupting. So which interactions or which bonds does urea or the guanidine hydrochloride break or affect, which leads to the denaturation of the protein? B, it's the hydrogen bonds. Yes. Mm, okay. So hydrogen bonds, I was a little bit thinking. So yes, so it is hydrophobic interactions, hydrogen bonds, and obviously the disulfide bonds, right? So yes, you have hydrophobic, hydrogen, and the disulfide bonds, because you have urea, it's a reducing environment, so disulfide bonds breaks. So ABC, no ionic interactions are affected. Okay. Now the next one is quaternary structure is best exemplified by. So one example of a quaternary structure protein: myoglobin, insulin, hemoglobin, keratin. So quaternary structures, the effect of cooperativity. Yes, it's hemoglobin, because there are 4 subunits that are required for the function of hemoglobin. Okay. And the last question, which are integral membrane proteins? Type 1, type 3, peripheral proteins, lipid linked proteins. Which are integral membrane proteins? No, peripheral proteins are not integral proteins. Peripheral proteins are very different from integral proteins. So if you go over here, what you have? You have type 1, you have type 3, and lipid linked proteins. That's type 5. So, yeah. So, A, B, sorry. So, yeah, A, B, and D. Okay. Yeah, I am done from my part. If you have any questions, you can ask me. Any questions, any doubts from this class, previous classes, anything? No questions? Okay. Thank you very much. We will again meet in the next week on Friday.